Okay.
Fantastic.
There
All
he
right.
is.
Well, he's the guy you want to talk to. So great to see you, Scott. I'm going to let you guys chat.
Hey, how's it going, Scott?
Doing pretty well. How are you, Tim?
I'm doing great. You got your sleeves rolled up. I mean, you're ready to go. Look at that.
Yeah,
He's
that's
working
right.
hard today. Hey, thanks for taking some time, being a part of this little radiothon that we try to do every year, helping raise funds, raise awareness. It's also a great opportunity to bring people in, such as yourself, to kind of raise awareness about what you're doing in your corner of the FSHD Society's group, community, and what's happening. How's the research going? People want to hear from you, I think, on that front for sure.
Sure. It's going well. So, we've talked before.
Yes.
Over the last couple of years about what we're trying to do, which is use gene therapy to treat FSHD.
Mm.
And, gene therapy is the sort of a basic definition of gene therapy is treating disease with DNA. So what we're trying to do in our case, probably talked about this DUX4 gene, this bad actor.
Of course. Yep.
We're trying to deliver a piece of DNA to muscle cells that can produce something we call a microRNA that will work to inhibit DUX4. And the way we've designed this is to actually use our cell's own machinery to produce this therapeutic sequence inside the cell. And the intention is to have it be a one-time treatment. So, we're delivering this then to, in our case right now, what we've done is we've delivered it to mouse models of FSHD, and we use a virus. So we package this piece of DNA inside a virus. We grow the virus in a lab. These viruses have a natural ability that they've acquired over millions of years to infect muscle. And one of the cool, so these are called adeno-associated viruses. We abbreviate those as AAV. And one of the cool things about AAV vectors is they can't replicate inside human beings. So they sort of deliver their payload, which is the DNA we're putting inside them.
Right.
The viruses go away. The DNA remains inside the recipient cells, which are the muscles. And then like I said, we just produce this therapy inside the muscle. And it's pretty stable. It'll last for years after one single treatment. So where are we then? We've done a lot of proof of concept studies in mice. So we know that this works. We know that this therapy can knock down or reduce DUX4, and it can have benefit in mice. We've actually seen if we deliver enough of this virus to hit enough muscle, we can correct most of the features of muscular dystrophy in these mice, including functional deficits. So we see the mice can't walk around a cage very well.
Right.
And so when we deliver these viruses with this therapy to these mice, we've gone out to, for a therapy study and a functional study, we've gone out to 10 weeks. And for some other studies, we've gone out to a year, and we're seeing correction out to a year after a single treatment. So the issue is that the viruses that we're using require very high doses to hit enough muscle. And so we're sort of at this place now where we are using this virus system, and it works great in mice, but the doses we think are probably too high for treating humans.
For human, okay.
So there are a number of other companies and academic labs that are working on engineering these viruses to allow you to be able to hit the same amount of muscle at much lower doses. And so our work then has been partnered, been licensed to a company that I'm actually a part of as a co-founder. It's called Armitis Bio. And Armitis has been helping us make some partnerships to try to identify the best virus system that we can use. We're calling these next generation viruses that we can use then to deliver this treatment to muscle at much lower doses, in a very safe dose range. And so we're testing a number of those right now. We have several we've actually injected a bunch of mice already.
Wow.
And we're hoping that by early January, we're going to have some good results on some of these newer vectors. And I think if we're able to show that we're getting the same sort of benefit with these new viruses as we are with what we call the first generation system, and we're able to do it at lower doses, then we're ready to take that jump and try to pursue a clinical trial.
So this information, is that going to be made public of what you're currently doing? You said in January?
Well, so this is actually a long study. The first group of mice that we've injected with these will be analyzed in January. And
Okay.
I think we'll have a good sense of if we have a lead virus system, a delivery system that we would be comfortable delivering to human. And then at that point, then we also, we have to still have to do safety studies.
Of course.
But we know sort of where the sweet spot is based on clinical trials that have been done for other muscular dystrophies using the same kind of virus.
Mm-hmm. So at this point, what would be the things that like Armistice Bio needs, or to kind of is there anything that the FSHD community can help in support of the work you're doing?
Yeah, so there are a few things. We're still raising money through the company. So these are gene therapy projects are expensive. Yeah. So we're raising money, but that's an issue that we'll have to deal with through the company. But also we are we want to seek we want to do what's called a lead-in study, right? So we want to have a group of patients that we can monitor and track disease progression before we actually begin the trial. So then we can have a better sense of the individual disease progression in a group of patients that might then be enrolled later on in our gene therapy study.
Okay. And so that's obviously community involvement. How do you, it's just similar to the MOVE study that we were just talking about before we had you on. You just kind of want people to be aware of it, and then will there be some type of registration and so forth?
Yes.
Just like theirs? Okay.
Yeah, eventually we'll get to that point. Right now, I think step one is to identify our lead delivery system, our lead virus. Step two is to prove that it's safe in animal models, and then we begin the march toward clinical trial.
Now, some might be listening and they're pretty excited because when you're talking about gene therapy, that's not pharmaceutical side of things, right? This is not a pill. You're talking about a virus is the delivery system for this gene therapy. So is it, this is of course a layman, I'm not a scientist, thank God. Is it then, would you consider it, I don't want to say safer, that's not really it, because it's just unique, it's a different pathway. But I get excited as someone with FSHD when you say it's like a one-time, thing. So we're talking like injection, you show up when this is, available. Hopefully that is what is coming down the pipeline, and that it's a one-time treatment. That's what the hopes are for, right?
That's the idea, yeah. So these viruses that we're using, they're pretty stable inside. Well, they are stable inside cells that don't naturally turn over very much. So for example, our skin cells, we're constantly
right.
Our skin cells are constantly dying, and we're constantly making new skin cells, right? So if you delivered a virus, because the key point here is that these viruses don't insert themselves inside the host cell DNA.
Right.
So they're not invading our chromosomes. They kind of sit outside our DNA.
They're just the delivery mechanism.
Exactly.
Yeah.
And so the therapeutic that they're delivering just kind of hangs around outside. So if a cell turns over, it dies and then it is replaced by a new one. The DNA that we've delivered from that virus goes away too. But in muscle doesn't really turn over that much unless it's damaged, unless it's diseased. And so we can deliver these viruses to muscle, and they will just hang around for an indefinite period of time. So we know, like a mouse only lives 2 years, and we can deliver this virus to a mouse, and we can wait 2 years, and it's still there 2 years later. I think the longest anybody's ever looked in a human being is about 10 years after a single treatment.
Okay.
But that's not to say that we don't really know how long they last.
Right.
In a human. But the idea is that it's not something that you'd have to get take a pill every day or an injection every
Mhm.
day. It's intended to be a single treatment that lasts a really long time.
That's exciting. I mean, as a patient side of it, that's exciting research. What made you get started within that research? Going in the Wayback Machine.
Yeah.
What was that motivation?
When I started my PhD program way back at the University of Michigan in 1996, 1997, I wanted to work on what we now call the type of work I wanted to do translational, but that term didn't actually exist back then. So what that means is, applied research. There were a lot of people at University of Michigan doing basic research, trying to understand basic biology. I wanted to do something that we could move, from a disease model in a mouse, for example, and try to move it to a human. So directly treating diseases. And I just happened to find a lab, like my PhD mentor was named Jeff Chamberlain, and he was working on gene therapy for Duchenne muscular dystrophy way back then. And that's sort of where I got my start, and I've kept with it. And we've just moved into different directions. I'm not working on DMD anymore, but we're still working on neuromuscular disease, and we're still using the same kind of gene therapy principles that I learned way back as a PhD student.
And you talked about, thanks for that part of it, as in motivated to do it, help our community get something started. Gene therapy, I think, is massively exciting. You talked about a little bit of the goals of what you see coming in 2023. So where do you hope to be this time next year?
This time next year, I hope we're wrapping up a toxicology study and recruiting patients for a potential clinical trial.
That sounds awesome. That's great. So that hopefully is going to be the theme of our conversation at the next Radiothon.
I hope so. Yeah.
That's what you're looking to do, and you're pulling in to get us to recruit for you and get some people involved.
That's right.
That sounds like a great goal. I like that. Well, thank you very much, Scott, for joining us, being here again on another Radiothon. I know this is becoming kind of an annual thing here with us, so I really appreciate your time.
My pleasure. I think I've told you this before. FSHD Society kickstarted my career as an independent investigator, and in particular, Dan Perez has been really supportive over the years. So, Dan, if you're out there listening, thank you again. Tell him thank you every time I see him. But the very first grant we ever got was for $10,000.
Nice.
From FSHD Society, and that really helped kickstart our work in FSHD.
Mm-hmm. Yes, it did. Yeah, it's good. It is good history, and you're making history as you walk forward. And I'm excited to see. I know the rest of the community is, to see where this goes for this time next year. I can't wait to keep following your research and the work you're doing, Scott. We really appreciate it. Thank you.
All right, thanks, Tim. Good to see you.
You too. I truly enjoy talking with Scott Harper.
I do too. I do too. He's such a down-to-earth guy, and he comes to the walk in Columbus every year, brings his whole research team. They bring a team of folks and come out, and I got the pleasure of seeing him and chatting with him there a couple of years ago at that awesome walk that the Aw Millers put on. Sue is our leader there, and actually Bob's not next, but the next two guests down the line. Scott's a great guy. And that's the power of this little tiny grant, right? He just got this small grant from the FSHD Society way back when, and look where he is now. I mean, what he's developing, should it be successful, from our mouths out to the universe, right? Like, if it is successful, that's a miracle drug, right? That's a miracle treatment that could just, stop it in its tracks.
Yeah, I was just blown away by the fact that you could get that injectable. I mean, that's the path. That's what they're hoping for.
That's what they're hoping for.
Yeah, I mean, I'm not trying to confuse people. No, it's not here yet, but this is where the work is going. This is where your funding is going. He mentioned it, that first, you said it also, that first $10,000, that was a grant. FSHD Society got them off the ground. And look where the research world is now. with it.
Right.
All leaps and bounds in the pharmaceutical company. So many more are getting involved with it. Scott pioneering his career, starting it off because of that. Going back to the theme of Giving Tuesday is important. Giving is important. Participation is important. That is the theme of the day here,
That's
folks.
exactly right. And we back in the day, years ago, a couple decades ago, it was all about small investigator grants to try and get people into the field. It was about how do we get researchers interested in researching in FSHD, because we got to move this along. So those small investigator grants were critically important to get the ball rolling and get things moving. We're in a different place now. Now we're at a place where when we put out a request for research funding proposals, we get more than we can fund. Good ones, not just, okay ones, like really good research proposals projects that we want to be able to fund. In addition to that, we need to continue to support patients in their everyday living through the educational offerings that we have through FSHDU and through the support groups, through the gathering place, Feeling Fit with FSHD, because that's a new one we launched this year. We're going to hear from one of our founding members of that later today. Yep. Doctors used to tell you, go home, don't move too much. We now know that's not the best advice. The right level of movement and exercise can help you maintain the muscle you do have, and that's important. And so this group has taken off. There's just so many things that need to be done, but we got to also keep funding progress and research. We got to get better biomarkers, like Neil was talking about. Jamsheed is also doing a project directly, our chief science officer, working on that and trying to identify those biomarkers. There's a lot that needs to be done. That all takes money. None of it happens for free. I wish it would, I wish it did, but it doesn't. And so if there's barriers to progress, if there's hurdles we need to overcome, we want to figure out what those are, or we want to work with the other people, not just ourselves, but others to get it done. When we have more research, good research than we can fund, we don't just let it die in the vine. We go talk to the Chris Carino Foundation, to Friends of FSH, to other groups out there and say, hey, we've got this great research proposal. We don't have enough to fund it. What do you guys think? Will two of you come together and get it funded? Because we're leveraging those relationships. It's all about moving things forward.